Okay, I'm going to introduce Eric Mallack and Christina Mertz. They are going to give, I guess, a dynamic dual talk on novel brain iron imaging. So Eric Mallack is an assistant professor of neurology at Johns Hopkins and Kennedy Krieger, and is an early stage NIH investigator who's focused on cerebral ALD. Christina Mertz is a very talented PhD assistant professor also at Kennedy Krieger and Johns Hopkins, and is building both mouse and human iPSC-based model systems to understand disease mechanisms. And they're going to give us an update on some of the work they've been doing on brain iron imaging and cerebral ALD. Thanks.
All right. Thank you. So thank you for inviting us. I don't see Dr. Lund, but every meeting I have the charge of doing something new to try to impress him. So hopefully he finds this more impressive than the last. So what we're going to talk about is some work that has been done in imaging. I've presented a little bit of this data at prior talks, and it's sort of matured over time. Looking at a novel imaging method that has given us sort of insight into potentially some new mechanism of disease. So I'll do the first part, the imaging part. I'm going to talk really fast, show a lot of pictures, but Dr. Nemeth, Mertz, her piece is the really important piece of this talk. So when we think about imaging, especially sort of going forward, where our collective heads are at is really thinking sort of beyond the lesion. So what's happening in other parts of the brain, and trying to reconceptualize cerebral ALD both in peds and adults as maybe something that's a whole brain event. We want to look at imaging modalities that can tell us something about the molecular pathogenesis of disease. But I'm a clinician by training, so I sort of try to fake it as a researcher. But I always come back to, well, what's actually clinically useful? I think a lot about feasibility and reproducibility across sites. So trying to be in that Venn diagram of finding something that's going to give us insight into disease, but also really be useful in the clinic. So. I'm going to talk a little bit about this thing called quantitative susceptibility mapping source separation. So this is an MRI technique that looks at the local magnetic field of all the species, all the molecular species in your brain when you're stuck in an MRI scanner. And so just to give a quick overview, when anybody gets an MRI, their brain is exposed to a magnetic field. That's sort of how we generate the images. But there are some species in the brain that, when exposed to this magnetic field, will locally upregulate or locally amplify that magnetic field at essentially a certain frequency. And in this case, one of the most prevalent species in the brain is actually iron. We accumulate iron in our brains as we age normally. So iron is really important in oligodendrocyte metabolism. And it's important in microglial activation. So iron is something that we call it paramagnetic. It upregulates it amplifies the local magnetic field when the patients are in a scanner. Conversely, myelin is diamagnetic. It's zippered closed, has almost no external electrons, and really repels the external magnetic field. So paramagnetic iron, diamagnetic myelin, one is responsive to the magnetic field, and then one is really not. And what we're going to do is look at perturbations in essentially iron and myelin. So this is what a QSM map looks like. It is the sum of both of those iron and myelin signals for all intents and purposes for this talk. But what we can do is take that and actually source separate it into effectively a myelin map. So what is the sum? What is the magnetic susceptibility of myelin, and can we put that into something that looks like an MRI? And then the QSM positive, which is essentially the iron map. So QSM negative, diamagnetic, myelin. I'm going to use the term sort of myelin throughout the talk. And then on the right we'll map out our iron. So we did this in boys, and we generated an atlas, an age match atlas, 5 to 10 years old, 50 patients. And then what we did is we mapped patients with cerebral ALD onto this atlas. So I'm going to walk through this pretty quickly. In the top row here, here's a patient who has a demyelinating T2 lesion. It's enhancing. This column here is the myelin map, and then this column here is the iron map. Red is elevated, blue is low, and green is somewhere in the middle. Second patient here, here's a patient with a lesion. It's non-enhancing, so they fit the criteria of self-halted disease. Again, myelin map, iron map. So I'm going to sort of break this down a little bit, but what you can really sort of take off the top, and the red and the blue are standard deviation changes. That's just sort of how we quantify it. Is in the myelin map, you can see the lesion pretty well. And then in the iron map, in the patient with progressive disease, there seems to be just out in the normal appearing white matter out here, way away from the lesion, an elevated iron signal. In the boy with arrested disease, not so much. So these are sort of initial observations. So if I zoom in and I look just at the iron signal, here is the T2 lesion. Here's the overlay of the variation, the standard deviation in iron concentration. You can see within the lesion, there's low iron, but there's these little islands here within the lesion where there seem to be pockets of iron. At the border of the lesion, low, and then in the normal appearing white matter, it's elevated. So you sort of overlay these two, and this is kind of what you get. And you can start to co-localize where you see the variation in signal. And I've sort of pointed this out before. This layering effect looks a lot like prior work done by Dr. Eichler looking at microglial apoptosis. And so this is just an initial observation. This is not a conclusion by any stretch of the imagination, but this is based on the imaging, the road we started to go down based on our findings. Looking at, is there variability in pathology that we know exists based on histopathology? Are we really capturing that on imaging? And I want to bring up this idea of gradient of pathology from normal appearing white matter to perilesional white matter to lesion and understand what those spatial changes look like. One point here is the QSM signal is dynamic. So here's the same patient 4 months apart. You can see this is the iron signal here 4 months later. It moves. Again, an initial observation. So it's something that's dynamic and actually changes with the patient over time. So to go back here, just to look at the myelin signal in this patient in particular. So what we see here is a tiny T2 demyelinating lesion. And then on the myelin map, we see this little drop-off in signal that appears to be larger than the lesion. So here's the lesion here. Here's actually the area based on the susceptibility weighted, the susceptibility image. There's a drop-off in the myelin signal that is essentially larger than what we see on T2. So the observation here is it appears that the QSM image actually, the T2 image maybe is underestimating the amount of lesion we actually have, and we're seeing it in here. So I came up with I didn't come up with this term, but this term penumbra is used, especially used in stroke neurology. Patients who have a stroke, you have the area of the stroke, and a lot of stroke care is trying to save the normal appearing white matter, at least initially when you have a stroke, the normal appearing white matter around that stroke so that injury doesn't continue to spread. So that's where this idea comes from. So here's the lesion. The QSM approximates the myelin signal drop-off to be actually larger. So you have this sort of, like I said, penumbra in here where even though this is normal appearing white matter on the T2, it's probably affected. So I put here, it appears to match the spatial distribution of lipid peroxidation. It was hard to tell this story because a lot of things happen in parallel, so just sort of take my word for it. There is 20 years worth of literature that shows it, and then Christina is going to show you some new data. But again, taking back to this idea of gradient of pathology, what is the imaging sort of telling you about the molecular and cellular events that are happening in real time? And then there's this signal out here. So this is an increase in the myelin signal. I don't know what it means, but this is after the patient fills out this area of demyelination, this is where the lesion's going next. We haven't really thought about how to account for this increase in myelin signal. Okay, so those were kids. some of the new work that we've worked on is actually doing this in adults. So same exact methodology. There's less control, so the data is going to look a little grainier. But again, what I'm going to do is sort of relatively quickly kind of go through some of our initial impressions. So, and I should say one more thing. I'm going to go through 3 patients. A patient with very minimal disease, so minimal projection fiber disease, non-enhancing. This patient is minimally symptomatic, has some complaints of stiffness in the morning, but walks around, has a job really, really early in their disease. Then the next step up, we have a patient who has your classic lesion. It's non-enhancing. This patient has AMN and has trouble walking, spends time in a wheelchair. And then the third patient has fulminant adult cerebral ALD with blood-brain barrier breakdown. So trying to get a sense sort of across the spectrum of severity. So here's a patient with minimal projection fiber disease. What we're going to show you here is here's the T2 flare. You see these hyperintensities that are really subtle in here. These are demyelinating lesions in the internal capsule. And what this is pointing at is, like I said, it's a little messy because we don't have a lot of controls yet, but what you can see here and here is that just like in the kids, the myelin map can pretty well localize to where the lesion is. So this was interesting. The patient doesn't have any lesions on T2 up in the centrum semiovalli near the cortex. And what you can probably appreciate is here's the control QSM that we generated from a bunch of adults. And then here's the patient. And precentral cortex in the iron signal is pretty bright. This hasn't been described in ALD before, but it has been described in ALS as a biomarker. So in ALS, they see elevated iron signaling in the precentral cortex. So this is a reproduction of something that we see in another neurodegenerative disease. Like I said, the heat map's a little messy, but you can kind of get a sense it's right here. This will become much more refined as we get more healthy controls. Again, the same patient, minimal projection fiber disease, but what we see here, interestingly, is a decreased iron signal in the pulvinar nucleus. Again, this is normal appearing tissue. So here's the patient, here's the thalamus, both sides. Back here is the pulvinar nucleus. One thing I wanted to point out, as I mentioned early on, is here's healthy adults. We all accumulate iron, especially in our deep gray matter, over time. Here's the pulvinar nucleus in healthy controls. And then in this case, for this patient, has what appears to be normal to maybe low. You can appreciate the blue here. But also for this patient's pulvinar nucleus, there's a dropout in the normal iron signal on both sides. So bringing up some of these sort of initial observations, one of the pieces that has been really enlightening in doing this sort of exploratory work is thinking about parts of the brain that we don't usually talk about in ALD and cerebral ALD specifically. Okay, so here's a patient with, this is another adult patient, late 30s, early 40s, has a splenial lesion, non-enhancing, and again, in adults, the myelin signal nicely approximates where we see it on T2, so it identifies the lesion. Non-enhancing, we looked back in the precentral cortex in that normal appearing tissue, and again, here's healthy control. You can kind of see that elevation in precentral cortex tissue, and it looks normal here. And it's a little bit elevated in here, but you get it better on the black and white here, that the signal in the omega sign of precentral cortex is elevated. And then lastly, we went back and looked at the pulvinar nucleus in this patient who is much more severely affected. And actually, interestingly, here's normal, or here's the healthy controls. For his case, we actually see it and it's elevated. So, I think this might be out of battery. Is there a pointer on here? I might have to just throw something at the screen. Okay, here we go. So, but you can see for this patient in their pulvinar nucleus that there seems to be an elevation in the signal versus healthy controls. Now, whether or not that this is a little bit of statistical noise, which may be possible, but if it correlates with disease severity, it would be interesting to look at over time. All right, and so lastly, we have a patient here who has pretty extensive cerebral disease, and again, just to show you that this myelin map can really identify the cerebral lesion. As you can see down in the cerebellum, so the bottom right corner of both figures, this patient has cerebellar disease, so white matter changes in the cerebellum, and again, we see that on the myelin map. Went back and looked at the pulvinar nucleus. This patient, again, this is a patient with advanced inflammatory disease that's actively moving. He has elevated signal in his pulvinar nucleus, just like the prior patient who had pretty advanced disease. And then sort of the last thing here is, again, as I mentioned, we accumulate iron. This is healthy control. We accumulate iron in our dentate nucleus over time. This patient, in the center of their disease, the iron signal has fallen off. And you can see that pretty well in the bottom left corner. So there's a lot here in terms of these observations to tell us a little bit about circuitry, to tell us about metabolism, to tell us about the network that may be affected. The thalamus is a major relay station for a lot of networks in the brain. So it could tell us a little bit about network physiology. So a lot of this is pretty exploratory, but at the outset, what I wanted to start to do is start to put this story together. So, it's 20, 30 years ago, it's sort of been known that in ALD in general, and cerebral ALD certainly, that oxidative damage plays a role in the pathogenesis of ALD. Lipotoxicity from oxidative stress. We're going to talk a little bit about this paper right here, oxidative stress of mice and men, but that there's redox imbalance that errs on the side of oxidative stress in ALD, so there's an imbalance there. And I'm starting to think, well, how do I put all this kind of together? This is work from Aurora Pujol, and she came up with this sort of three-hit hypothesis of how do you get from increased very long-chain fatty acids to oxidative stress to neuroinflammation. And I think the story that we're starting to hone in on lives kind of somewhere in here. So this is a paper, it was published 20 years ago. And just in the highlighted areas, I'm just going to not read it, but summarize for you. Ferritin was observed within oligodendrocytes beyond the demyelinating edge. H&E, which is that second paragraph, that second sentence, is a stainer, is a marker of lipid peroxidation. That was detected within the demyelinated lesion and outside of it. And if you look at that last line, this was really stuck out to me when going back to this literature 20 years ago. I'm going to read this. Lipid peroxidation is particularly germane to a rapidly progressive disease of myelin because once lipid peroxidation begins, it self-perpetuates and doesn't normally stop until the target substrate is gone. So that sounds like this sort of rapidly progressive disorder that we're dealing with. So the question is, how do we put this all together? We know that there's early oxidative stress in ALD. There's lipid peroxidation. I'm getting an elevated iron signal on the imaging. I'm guessing that in the myelin signal, that change in myelin susceptibility is probably due to lipid peroxidation. And then from Dr. Eichler's early work, we know that there's microglial activation in the normal appearing white matter, and as you transition to the lesion, there's perilesional microglial apoptosis, which is an early pathogenic finding. So we started thinking about this idea of ferroptosis, lipid peroxidation in the setting of iron. This is a new pathway for cell death, and it's been studied now in pick your neurodegenerative disease, pretty much every single one of them. And this is a fancy slide. Essentially, it's just saying when lipids become peroxidated, they go from being tightly packed together. These peroxide molecules have charge on them, and they start to break apart the lipid membrane. And I think that's what we're picking up on the magnet. We're making myelin more susceptible by adding anions and cations to it. So I'm going to hand it over to Dr. Mertz because she's done some really, really interesting work trying to validate some of these imaging findings in the basic science lab. And I kid you not, when I presented a lot of this data to her, we ended up going to the basement of Kennedy Krieger and pulling out brains from the 1980s and started staining them to start to look for cellular molecular evidence of our hypothesis. So.
Hi, thank you. I just wanted to start by thanking everyone and saying we're really excited to present this work and get everyone's sort of feedback on it. Yeah, so let's talk about ferroptosis a little bit. So Eric sort of set the stage for it, but it is this iron-dependent programmed cell death, first described in 2012, but we really think that Jim Powers, Kirby Smith, Moser paper really sort of hit the nail on the head 20 years ago with this. So it's really, the whole thing about ferroptosis to me, I think, is this like balance of sort of everything. And so for first and foremost, this oxidative state, so pro-oxidative and anti-oxidative processes, and then of course the three hallmarks of ferroptosis, so iron dependency, dependency of that antioxidant balance, and then lipid peroxidation and polyunsaturated fatty acid incorporation into membrane phospholipids, which really kind of facilitates that peroxidation. And so, different lipids, and Nadav really eloquently went through all these different lipids. Each of these has a different sensitivity or a different susceptibility to being peroxidated and to undergoing ferroptosis. And so we really think that there's a lot of ways here for ALD to kind of be susceptible to ferroptosis. And so just touching a little bit on some of these different lipids, arachidonic acid sensitizes cells to ferroptosis. We certainly know that's involved in ALD. And then something that I thought was kind of interesting, 7-dehydrocholesterol, so this is a precursor to cholesterol and to vitamin D, is actually protective against ferroptosis. So just a balance of all these lipids within the brain really makes a big difference when kind of thinking about some of these cell death processes. And then, related, so I think a lot of us who think about the mechanisms of ALD on a daily basis can really see sort of these axis points of ferroptosis in the setting of ALD. So obviously we have a loss of antioxidant defenses. We have this very long chain fatty acid accumulation that both results in oxidative stress, but then throws off that lipid balance. And so there's a lot of places here for that to really take place. And kind of focusing more a little bit on iron is these, every cell in your brain needs iron. It's very important. And so when your microglia do not function properly, now you have a failure to maintain that iron homeostasis. And then we know that under conditions of under conditions of inflammation, the iron is actually sequestered as sort of a protective mechanism. And the more iron that's sort of held in the cells, that leads to more neurodegeneration. That's toxic for neurons over time, which leads to more inflammation. And the whole idea of ferroptosis to me also is this sort of feed-forward rapid progression and rapid degeneration. And so to really look at this, we did start with brains in a freezer in the basement, but we sort of went a little bit more legitimate after that, and I contacted the NIH Neurobiobank. And so I really had the honor of working with this brain tissue, and so we got samples from 3 ALD cases. I have 2 shown here, and then 1 control. And so I just want to talk a little bit, the limitations, of course, of working with human tissue. It's amazing, it's a wonderful resource, but we don't know anything about these cases. And so what I wanted to do is I wanted to get brain tissue that we thought was certainly lesioned, and then brain tissue that might perhaps have some normal appearing white matter so that we could explore some of these mechanisms. And that's hard to do when you don't know anything about the cases. And so I picked inferior parietal lobe, which is what we're seeing here. So 2 of the 3 ALD patient samples had inferior parietal lobe, and that's pictured here alongside a control. Not age matched, unfortunately, but I think if we're looking at iron, that's sort of an okay thing since iron accumulates with age. And then what I did is just sort of basic histology. So first looking at myelin, which is what you're pictured here. So this is Luxol-Fast blue and a Crescel violet counterstain. And then I looked at lipid peroxidation and then, of course, iron. And so you can see sort of 3 varied cases. Obviously, we have more myelin loss in this 10-year-old compared to the 25-year-old, but I think each of these sort of presents an interesting case for us. And I'm going to start right with the iron. So this is Prussian blue, and this was a really simple stain. And when I saw these tissues, I immediately just texted Eric frantically because this is much more iron than we sort of expected. And so these lesion sites, this is actually from the periventricular white matter from each of these cases, and this is the 17-year-old that I didn't have on the previous slide. But what we see in each of these cases is significant iron deposition, and the pattern is interesting. It's different in a lot of these cases, and that probably has to do with each individual case, of course. But in the 25-year-old, within this periventricular white matter, it was sort of clustered. These sort of look phagocytic cells. There might be some accumulation within blood vessels. This 17-year-old was a very severe case. Here it seems to be much more diffuse and spread over a wide area. And then with the 10-year-old, this tissue was actually quite necrotic, and for a large swath of this tissue, it actually doesn't stain for nuclear fast red. And I think what we're seeing here is a lot of debris that's actually spilled out of the cells, but we do still see iron that's sort of accumulated within the tissue. And then if we look in perilesional areas, we see sort of a different story. And I think what we're seeing here is also fairly represented well in that Jim Powers paper, where we do see very diffuse iron within these perilesional areas. And so here you can see disrupted myelin, disorganized myelin. And you can see I wish I could zoom in because it's one of these things that the more you stare at it, the more you see. But we do see diffuse punctate iron that doesn't seem to localize to a particular cell type, but it seems to be sort of everywhere. And I've done a couple of counterstains to explore that further. Same thing with this perilesional area within the 17-year-old. You can see the myelin here. Again, is a little bit disorganized, and you get sort of a different pattern of iron, which it might be accumulated there within certain cell types. And then this pointer's dead. But in the 10-year-old in the perilesional space, we actually saw less myelin, but it's still there. You see these blue punctate spaces, again, within that disorganized myelin. So now looking at lipid peroxidation. So what I try to do here is to capture sort of the gradient or these transition areas to use each patient sort of as their own control so that you could observe the changes over time. And what you see is a difference within these within the pattern of MBP. So MBP is in green, so here we're looking at myelin basic protein. You can see sort of organized or compact myelin. I have a small box up there in the top that shows where that is. That's kind of in the gray matter that's bordering the white matter, and there's a slight region of demyelination. But when you look at this tissue, you see within the perilesional white matter, the myelin is starting to get disorganized, it's starting to break apart. And then you reach that lesion area where we have a loss of cell identification of markers. But you still see that the marker for lipid peroxidation, that 4-H&E, does stain in some of those fragmented myelin bits. Similar with the IBA1, in this case, you can see the transition of somewhat ramified healthy microglia towards the bottom, and then that changes over time. And I think that change in microglia morphology is much more pronounced in this patient. And you can see the H&E here as well, where you can see the lipid peroxidation does co-localize with MBP quite a bit. Even though this patient seems to have a much more severe demyelinative or lesion area within that tract, I think it's not represented when we get 60X in that patient. And then just looking a little bit more high mag at these same images. So we see, again, the H&E pattern with the IBA1, the microglia. And again, you see within the 10-year-old patient, the lipid peroxidation within the myelin sometimes co-localizes with the microglia when it hits these sort of vacuoles almost that's within the lesion area. And then the bottom row, I did a marker for ACSL4, and this is actually an enzyme that sort of primes lipids for ferroptosis. So it helps to incorporate these polyunsaturated fatty acids into the lipid membrane, and we do get significant staining for that throughout the tissue, both in the lesion and the perilesional areas. And what's interesting is out in the gray matter, it seems to really be in neurons, which I think is exciting and something that we can explore a little bit further later. There we go. And yeah, so I think that's it. Thank you very much for listening, and we'll take any questions.
Questions? Okay, Florian.
Wonderful work, and it's so nice going back and forth between MR imaging and autopsy tissue. I guess the connection between iron metabolism and ferroptosis makes more sense to me in the inflammatory demyelinating scenario with necrosis and cell death. What I don't understand is what's happening in the motor cortex or in the thalamus, where we don't think of any primary cell death happening. And yes, there are tracts, such as the corticospinal tract, and sensory pathways that end up in the thalamus that you could imagine are tracts under duress, with maybe the soma in that at the end of that tract being affected. But yeah, I'm trying to understand how you would solve that, and whether you said you had seen this in ALS before. Is it really? Maybe I'm wrong. Maybe it's not about the neuron and the soma. Maybe it's microglial activation, as in ALS people have assumed. So I'd love to hear your thoughts.
So thank you for the question. So this is the nice part about having the opportunity to build a new team and really collaborate. So the people who are sort of listed on our last slide, a couple of them work at Hawkins and our Kirby Center, and they work in other diseases. And this was sort of brought up by one of the PhDs. Dr. Shin brought this up, and as he was scrolling through, he goes, "Oh gosh, we see this in ALS." So that was last week. I need to do my homework and start to read up on what they think that signal means. But I think what you're saying is sort of what we're thinking. We have these long white matter tracts that are affected, but we have these distal cell bodies sort of in cortex, in thalamus. Are they changing their metabolism and sucking up iron because their downstream axons are under duress? And are we seeing that? And that's the reason why I brought up that it's 3 observations, right? But the one patient with inactive disease who's walking around and doesn't have progressive cerebral demyelination, the pulvinar nucleus in his thalamus is not doing much. But the 2 patients with active disease, especially the one with active inflammation, is that a metabolic change that we can pick up on imaging to signal to us that maybe this patient is at risk for progression. So, and then it hopefully tells us something about the biology. So the long way of answering that is I have to go learn about what this means in ALS, and then take some of those lessons. And then we can start to think about it at the bench, how to test for changes in iron metabolism. So I think we're really at the basement floor here and pretty excited to kind of take this as it goes.
Other questions?
You say that this is a novel brain imaging thing, ironing, and so just interesting. So like, I've been in MRIs, and MRIs pretty much stand still, and you just hear all the noise or whatever, and then you just, sometimes you have a dye that the doctor, the MRI tech that he gives you. But how is it so different from a regular MRI machine that we usually take, that we're just in there and we're just hearing the noises and standing still and waiting for the image to come out for the doctors to see?
Yeah, that's a really good question. This is actually a similar sequence, a similar type of MRI sequence to the other sequences that you get. So one of the standard sequences we do is something called susceptibility weighted imaging, and that traditionally looks for blood in the brain. Well, the reason we do it in ALD or as clinical standard of care is you get the susceptibility sequence, you scroll through and say, okay, there's no blood in the brain, or there's not blood anywhere where it needs to be. But that signal that it looks for is actually iron. So this is a cousin to that type of sequence. It takes 4 to 6 minutes to acquire. It's short. So this is where I start to marry the idea of and really pull in the idea of something that's clinically useful and feasible.
Thank you so much. Absolutely excellent presentation. And these 3 cases are very, again, definitely a novel imaging modality in ALS or in ALD space and also in ALS. My question is how to relate this to the clinical symptoms and what is the objectives? Because, of course, it's absolutely important to describe different locations of iron accumulation in various locations in the brain, but what is the clinical meaningfulness of these findings and how to relate maybe to the findings that cannot be identified just by 6 minutes walk test or by time up and go, because obviously ALD is probably much broader disease affecting other functioning. And so how do you see that using of this novel imaging modality can help to predict other, for example, neurocognitive dysfunctions or problems? Thank you.
Yeah, so there's a great paper that came out of the Minnesota group by Dr. Pierpont looking at DTI and neurocognitive outcomes in ALD. And I think some part of the emerging picture is, we've focused so long on the lesion, right? This is the thing we're trying to stop. It gets really big. But now that we are identifying lesions early and treating them early, we see also these more subtle phenotypes in patient, neurocognitive symptoms essentially not accounted for by the lesion itself. So it tells you that something else is probably going on in the brain. In the adult population, again, the second patient I showed, he has a lesion in the splenium of his corpus callosum, but he certainly has neurocognitive deficits that I would not ascribe to just the area of demyelination. So, the clinical sort of utility I think is multiple-fold. There's the pattern recognition. Can this parse phenotypes? Then you start to get lesion versus non-lesion part of brain. What can this tell us about non-lesion part of brain? Does the signal severity differ between phenotypes? Can this help parse out the VUS cases from real pathogenic cases? You'd expect that the signals should be really close to controls in patients with a true VUS that's likely benign or benign. So there's the clinical utility piece there. And then you start getting more complex, right? So the thalamus also is a relay station for your limbic system and your emotional system and executive function. So you can start to become more sophisticated in your imaging investigation, starting to think about first and second order networks. And then, then the idea is bring it back to the basic science level to actually tell us something about the underlying biology. So I think the clinical implications are big, but bear a lot more study. But I like where the field is going and starting to think about the ALD brain in its entirety and allowing us to sort of move past just the T2 demyelinating lesion. Yeah.
Eric and Christina, I see a few more questions in the audience, but we're tight on time. We're a bit over. So maybe we'll stop here. I've got some thoughts for you, and I think there's some in the back here who might have some questions as well. But thank you very much for great talk.